in the reproduction lesson, how many pages we have discussed to this moment? Please check it out. Sexual reproduction. Page number
6.
6. Page number 6 mean I think I have completed up to this, correct?
Yes, sir.
Right.
Yes, sir.
So we are going to hereafter discuss individually male and female reproductive systems. Here, the picture depicts you a male reproductive system. I will first indicate you the components of it. Take a highlighter and highlight the components of male reproductive system. Okay, there is two testes there. It's a part of male reproductive system. On the testes, there is a coiled tubular structure called epididymis there. From the epididymis, there is a tube going vas deferens. In a male, there will be two testes, two epididymis, two vas deferens. This vas deferens travel and finally join this unit. It's a part of the male reproductive system, seminal vesicle. Further, there is a gland, prostrate gland. Gland is halfway, you have to complete it. Prostrate gland. Another gland, bulbo-urethral gland. Urethra, penis, scrotum. All these are male reproductive system parts. All these are parts of male reproductive system. So what are the parts of the male reproductive system?
Epididymis, scrotum, testis, penis, urethra, vas deferens, prostrate gland, seminal vesicle,
bulbo-urethral gland.
That's all, no, sir.
Told everything.
Told the parts you told to highlight.
But I heard only half. Anyway, so these are the parts of the male reproductive system. In this, as I told you already, how many testes are there?
2.
2 testes are there. How many epididymis are there?
2.
2 epididymis are there. 2 epididymis are there. How many vas deferens are there?
2.
2 vas deferens are there. Seminal vesicles, they are also, how many are there? 2 are there. Prostrate gland, Prostate gland only, what is it?
One.
One is there. Bulbourethral gland. You have, what is it? Two bulbourethral glands there. Okay. And urethra, one there. The penis or penis is also, what is it? One there. This is how they are divided. Could you please mark it down?
Done, sir.
Right. So we are going to work on each of them. Before that, here they are telling what are the gonads in male reproductive system. Which one are the gonads of the male reproductive system?
Testis.
Testes. What are the functions of the testis?
Produce sperm
Hmm.
and reproductive hormones.
Reproductive hormones. Thereafter, epididymis is there. What is the function of epididymis?
Store mature sperms.
Mature sperms are stored in the, what is it? Epididymis. Actually, in the epididymis, sperms will become mature. Thereafter, they are stored. What are the accessory glands in the male reproductive system? Mean three glands are there. Seminal vesicles are glands. Bulbourethral glands are there. Prostate glands are there. Okay. So in this, other than epididymis and gonads, there are glands there. There are ducts there. Urethra is a duct. Vas deferens is a duct. There is another duct called sperm duct. I will later show you. Sperm duct. So, what are the gonads in the male reproductive system?
Testis.
Testes. What are the accessory glands in the male reproductive system?
Seminal vesicle, bulbourethral gland, prostate gland.
Prostate gland. What are the ducts in the male reproductive system?
Urethra and vas deferens.
Vas deferens, urethra, and sperm duct. What is the third one?
Sperm duct.
Sperm duct. What is the function of accessory glands? What is the function of accessory glands?
Secrete products required for sperm movement.
Sperm movement. What is the function of the ducts?
Transport the mature sperm and glandular secretions.
Secretion. Right. If you take male reproductive system, the main component it is testis. Okay. In the outside part, from outside, you can observe two components only. One, you can observe the, what is it? Scrotum. Other one, you can observe the penis. Only those two structures in a male rather than finish. Right. So, if you take the scrotum and penis, they are the only two components that you can observe in the outside. Scrotum is a thin, skinny pouch. Scrotum is it's a pouch-like structure. Okay, a skin structure, a pocket-like. In the scrotum, there will be two. This is the scrotum. Here, it is divided into two compartments. In each compartment, one testis would be located. One testis in one compartment, other testis on the other compartment. Actually, what is the significance of it? Why? What is the benefit of, now for simple example, in female, we don't see any scrotum-like pouch. In male, it is testis mean. In the female, it is the ovary. Ovary is inside the body, but testis is situated outside the body cavity, inside the scrotum. The main reason here is when it is placed in the scrotum, the temperature in the testis environment will be 1 to 2 degrees Celsius less than the 1 to 2 degrees Celsius less than the body temperature. And that temperature is important for effective and efficient sperm production. If these testes are situated inside the body cavity, like in the female, then due to high temperature, that is 37 degrees Celsius in the human body, sperm production will be highly affected. That is the important idea. And in the notes, one more thing you are told. On every testis, there will be a epididymis. There will be an epididymis situated on every testis. From every testis, from every epididymis actually, a vas difference starts to go. Okay, for vas difference, here they are using another word called spermatic coats. Actually, the entire vas difference will not be seen inside the scrotum. Inside every compartment of the scrotum, one testis will be there, one epididymis will be there on the testis, and a vas difference will start and go. The initial parts of the vas difference can be noticed inside the scrotum. Okay.
Okay, sir.
Right. So, see whether you are clear about the scrotum. If you are clear about the scrotum, we will now discuss the testis.
Clear, sir.
Okay. Now we are going to speak about the testes. The first point I already told you, the significance of keeping the testes in the scrotum. A temperature about 2 degrees Celsius less than the core body temperature is maintained in the testis, and that is very much important to maintain the sperm production, okay, effectively. Otherwise, sperms will be deformed. If you consider the testis, in the beginning, like in the female, it will be formed inside the abdominal cavity. It will be formed inside the abdominal cavity. When the child is born, just before the birth, near to the birth, testis will be descending into the scrotum. The development site for the testis is abdominal cavity. In the female, ovary also will develop in the abdominal cavity. In her, ovary will not get down, but here testis will get down very near to the birth. Right. In the testis, if you observe the testis, in the testis, large number of small compartments will be there. Large number of small compartments will be there in the testis. I am going to show you a diagram. Right, I am taking this one. Right, these compartments are called by the name lobules. What is the name given to the small compartments in the testis?
Lobules.
Lobule. In each lobule, there will be seminiferous tubules. One lobule can hold 1 to 4 seminiferous tubules. Like this, in a single testis, 200 to 300 lobules can be there. 200 to 300 lobules can be there. In every lobule, 1 to 4 seminiferous tubules will be there. Understood?
Yes, sir.
Right. What is the TV sound? TV sound, radio sound. எப்பவுமே லேர்னிங் பிளேஸ் வந்து அமைதியான இடமா இருக்கும். Right. Look at this picture. This picture shows you seminiferous tubules in the cross section. It is showing you seminiferous tubules in the, what is it?
Cross section.
Cross section. Hafsa Shafi, where are you?
I'm here, sir.
Sattam.
ஒரு சத்தமும் இல்ல சார்.
கேக்குதே. சரிப்பா. Here in this picture you are shown how many seminiferous tubules mean. It is shown to you 5 seminiferous tubules. Understood? It is shown to you 5 seminiferous tubules. This is one C, like that. Here it is one. Here it is one. Here it is one. Here it is one. Okay? Understood?
Yes, sir.
Right. If you take one seminiferous tubule without taking many, if you take one seminiferous tubule, this one, basically there will be 2 types of cells in it. How many?
2.
2 types of cells in it. One type of cells are called germinal epithelial cells. What is the name given?
Germinal
epithelial cells. If this is the seminiferous tubule, immediately in the wall of the seminiferous tubule, okay, there will be germinal, what is it? Epithelial cells. So just I am marking like this. 1 cell, 2 cell, 3 cell, like germinal epithelial cells will be there. Understood?
Yes, sir.
Right. Further, and from the germinal epithelial cells, certain cells will be extended like this. These extended cells are called Sertoli cells. What are the 2 types of cells in the wall of the seminiferous tubules?
Germinal epithelial cells and Sertoli cells.
Germinal epithelial cells and Sertoli cells. Sertoli cells are comparatively large cells. They will be extended towards the center of the, what is it? Seminiferous tubule. So what are the 2 components making the wall of the seminiferous tubule?
Germinal epithelial cells, Sertoli cells.
Sertoli cells. Germinal epithelial cells can undergo meiosis, mitosis as well. What is it? Meiosis. Actually, they will undergo mitosis. Some cells will be made from them by mitosis. Those cells will undergo meiosis. Some cells will be made from them. In that way, finally, they will end on they will end on sperms. Clear?
No, sir.
From here, I will further show you that. From here, there will be several cells made. Understood? There will be a line of cells made. Some cells will be undergoing mitotic division. Some cells will be undergoing meiosis division. Final product of those divisions is a sperm. Understood?
Yes, sir.
Okay. Who will undergo meiosis? Who will undergo mitosis? I will further explain you. Okay. So, in other than germinal epithelium and Sertoli cell, anything you notice there will be different stages of the sperm production. Understood?
Yes,
Germinal epithelium, Sertoli cell. Except these, any other cell you notice inside the seminiferous tubule will be one stage of sperm production. If you look at this diagram now, with that idea, you will notice, in this region, various cells are given to you. Read the names of them. Spermatogonium. Can you see that word?
Yes, sir.
Spermatogonium. Primary spermatocyte. Then secondary spermatocyte. Then spermatid. Then
Differentiating.
Ahika, I think your sound one is sounding. ചർന്ന് ഒരു ഇരച്ചിലുണ്ട് വരുന്നത്. Right. Spermatic. Okay. And finally, it is called spermatozoa. That is the young sperm. Young sperm will turn to mature sperm in the epididymis. So, what are the stages of sperm production you will notice inside the seminiferous tubule? What are the stages of sperm production you will notice inside the seminiferous tubule?
Spermatogonium. Primary spermatocyte. Secondary spermatocyte. Spermatid. Differentiating spermatid. Spermatozoa.
Spermatozoa. Right. Sertoli cells. Now, can you see this yellow color portion is actually representing Sertoli cells. Okay. Do you notice spermatogonium, primary spermatocyte, secondary spermatocyte, spermatid, spermatozoan, all are attached to the Sertoli cell?
Yes, sir.
Right. That is because Sertoli cell is giving them nutrition. Okay. they get the nutrition they want from the, what is it?
Sertoli cells.
Sertoli cells. Sertoli cells have another talent also. Sertoli cells can secrete a hormone called inhibin. What is the hormone secreted by Sertoli cells?
Inhibin.
Inhibin. We will inshallah later discuss what is the role of inhibin in the male reproduction. So do you agree? In the testis, there will be many lobules.
Yes, sir.
Yes. How many lobules about I told you?
200 to 300.
200 to 300 lobules will be there. In every lobule, how many seminiferous tubules will be there?
4.
1 to 4. Even it can be least to 1 and highest to 4. Clear? Are you clear?
Clear, sir.
Right. The basic two types of cells in each seminiferous tubule are two. What are the two types of cells? What are the two types of cells?
Germinal epithelial cells and Sertoli cells.
In these two types of cells, cell can undergo cell division. In these two types of cells, which cell can undergo cell division?
Germinal
Epithelial cell. Sertoli cells will not undergo cell division. Right? So, in these two types of cells, who will contribute to develop sperms? Germinal epithelial cells. Right? From the germinal epithelial cells, the cells made are called spermatogonium. From the spermatogonium, the cells made are called primary spermatocytes. From the primary spermatocytes, the cells made are called secondary spermatocytes. From the secondary spermatocyte, the cells made are called spermatids. From the spermatids, the cells made are called spermatozoa. In these stages, which one you will call sperm? In these stages, which one you will call sperm?
Spermatozoa.
Spermatozoa is the sperm. What are the functions of Sertoli cells?
They provide nutrition and they secrete hormone inhibin.
They provide nutrition to all the developing stages of sperm. Spermatogonium also get nutrition from it. Primary spermatocyte also get nutrition from it. Secondary spermatocyte also get nutrition from it. Spermatid also get nutrition from it. Even spermatozoa gets nutrition from it. Understood?
Yes, sir.
Right. If you see the left side picture, among that means between one seminiferous tubule and another seminiferous tubule, there is a white color region. Can you see?
Yes, sir.
That white color region is called interstitial region. What is the name given to that region?
Interstitial region.
Interstitial region. In the interstitial region, now one kind of cells are there. What are the cells?
Leydig.
Leydig cells. Leydig cells are responsible in secreting the hormone testosterone. What is the hormone secreted by the Leydig cells?
Secrete testosterone hormone.
Testosterone hormone. So altogether, by the testes, how many hormones are secreted?
2.
2 hormones. What are the hormones?
Inhibin and testosterone.
Who secretes the inhibin?
Sertoli cells.
Sertoli cells. Who secretes the testosterone?
Leydig cells.
Leydig cells. Are you clear?
Clear, sir.
Right. Please read through this note and tell me. Are you clear?
Sir,
Yes, ma.
so what are androgens?
Androgens are male sex hormone like certain chemicals. Okay, they are not considered as hormones, but they also will develop certain what is it? Male characters. Now, okay, it's a kind of steroid hormone-like structure. Same time, now you have to have seen in some girls. Have you seen mustache growing?
Yes, sir.
Those things are due to androgens. Understood?
Yes, sir. Clear, sir.
Right. Now, it is time to discuss about the epididymis. Can you see the epididymis in this picture?
Yes, sir.
Yes. It is epididymis here pointed. There are three parts in epididymis. What are the three parts in epididymis?
Head, body, tail.
Head, body, tail. But I don't know whether it is mentioned to you in the notes. Anyway, your book says seminiferous tubules, they join together and make certain tubes, and they join and form the epididymis. Okay, but those names are not in your syllabus. Okay, aberrant ductule, efferent ductule. No, those things are in your syllabus. Just to show you how epididymis is featured, I picked this picture. Clearly showing, no, correct? This is epididymis situated on the testis, connected to testis. Okay, those ductules that are connecting the epididymis and testis are actually connecting the seminiferous tubules and the epididymis. Sperms made in the seminiferous tubule will be transferred to epididymis. Okay, and they are stored. They become mature. What does the note says you? Epididymis is a tightly packed mass that is formed by repeatedly folded long duct originated from combined seminiferous tubules in the testis. From seminiferous tubules, sperm will be carried to the epididymis. According to the notes, epididymis is how much long?
6 meter.
6 meters. So it is a very long one, correct? The total human body will be less than 2 meters. Our total length is, our body's length is how much? Less than 2 meters. Then 6 meter, considerably a longer length or not?
Yes, sir.
Through this longer tube, microscopic unit called sperm is going to travel. In point of view of a sperm, it is a very longer journey or not?
Yes, sir.
Right. So that's why your book is telling it will take how long the time for a sperm to travel from one end of the epididymis to other end of the epididymis?
3 weeks.
3 weeks. 3 weeks time it will take. Okay. When it is traveling, what is happening to it? Little by little, it's becoming mature. Remember, mature sperm only capable of fertilizing. Understood?
Yes, sir.
Further, the sperms reaching the epididymis from the seminiferous tubule are not mobile, cannot move. So within the epididymis, they are becoming, what is it? They are gaining their mobility, right? So until it is ejaculated out of the male body, sperm will be stored in the epididymis. Understood? Now after epididymis there are parts, there are no vas deferens. Okay, many other parts are there. No? In none of them sperms will be stored. Mature sperms will be stored in the epididymis. The only thing that can make the matured sperm leave the epididymis is ejaculation. Okay, releasing the semen out of the male body. Understood?
Yes, sir.
Now, we are coming to this vas deferens, ejaculatory duct, urethra, penis. Regarding them, it's not good ideas there. Can you see the urinary bladder here?
Yes, sir.
Yes. Can you notice vas deferens? Vas deferens is coming here. Can you see?
Yes, sir.
This vas deferens, can you notice in this place joining with the duct of the seminal vesicle?
Yes, sir.
After joining, can you notice a single small duct is formed? This is the duct of the seminal vesicle. This is the vas deferens. After they bind, can you see they form a canal here?
Yes, sir.
And what is the name of that canal?
Ejaculatory duct.
Ejaculatory duct. So small duct there. Clear?
Yes, sir.
So who are the components joining to form the ejaculatory duct?
Seminal vesicle.
Seminal
And
vesicle. You say
semini.
duct of the seminal vesicle. You say duct of the seminal vesicle and vas deferens. Who two join together?
Vas deferens and seminal.
Seminal vesicle. So ejaculatory duct is not a bigger one, it's a smaller one. Immediately ejaculatory duct open to this red color portion. What is that red color tube?
Urethra.
Urethra. Urethra beginning part is surrounded by whom?
Prostate gland.
Prostate gland. From the prostate gland, tiny, okay, tiny tubes will come and open in the urethra. Understood? Now for seminal vesicle, okay, for seminal vesicle, there is a duct, right? For this one, there's no single duct. Tiny, many ducts are there. All the ducts are opening in the urethra. Understood?
Yes, sir.
Right. When you go down, you will see the urethra. In the urethra, another two glands are opening. Okay, they are directly opening. So what are the glands opening to the urethra?
Prostate gland and urethral gland.
So seminal vesicle doesn't open to the urethra. Seminal vesicle opens to the seminal vesicle opens to the
ejaculatory duct.
Ejaculatory duct. Are you clear?
Yes, sir.
Good. So during ejaculation, sperms are propelled from each epididymis through a muscular duct. So they are describing vas deferens as a muscular duct. Each vas deferens extends around and behind the urinary bladder. Behind the urinary bladder, it joins the duct from home. Duct from home. Seminal vesicle. Those two together will form a
ejaculatory duct.
Ejaculatory duct will open into the urethra. Okay, urethra is a common passage for
urinary
excretion and delivery of sperms. Sperms will not individually come there. Sperms will be coming as a mixture of semen, and in natural reproduction, the semen will be introduced into the female reproductive tract. Okay, right. Urethra runs through the organ called penis. It opens to the outside at the tip of the penis. In the tip of the penis, there are erectile tissues. Okay, penis is made of what kind of tissues? Penis is erectile tissues. In the penis, in the erectile tissue region, okay, erectile tissue contains modified veins and what is it?
Capillaries.
Capillaries. Okay, no need to go for lots of explanation on that part. If you just get to know, that is much more enough. Clear?
Yes, sir.
Right. So please go through this heading, okay, and tell me whether you are okay with it. If you are okay with it, we will go to the next one.
Clear, sir.
Okay, clear?
Yes, sir.
So if you are clear, what is our next heading?
Spermatogenesis.
What is spermatogenesis?
Male gamete formation.
How it is the definition of spermatogenesis? Long definition. It is the male gamete formation. In it, what are the steps included? Number 1, read it. Formation of spermatocytes from spermatogonium. Have you heard about the word spermatogonium?
Yes, sir.
Spermatogonium will produce which spermatocyte?
Primary.
Primary spermatocyte. That primary spermatocyte is the one that undergoes meiosis 1 division. At the completion of meiosis 1 division, okay, what would be the outcome? Who will be produced?
Secondary.
Secondary spermatocyte. It is the secondary spermatocyte that undergoes second meiosis 2. From secondary spermatocyte, who will be formed?
Spermatids.
Spermatids. Now, one primary spermatocyte, if it completes meiosis 1, how many secondary spermatocyte you guess to be produced? One mother cell completing meiosis 1, how many daughter cells will be produced?
Two.
2. Meiosis, that is meiosis, right? One mother cell undergoing meiosis 1 will produce 2 daughter cells. Each daughter cell will undergo meiosis 2. Now, how many daughter cells will be produced at the end of meiosis 2?
4.
That means from one primary spermatocyte, how many spermatids you are expecting?
4 spermatids.
4 spermatids you have to expect. Okay, you are pointed. The time taken to produce mature sperm from a specific spermatogonia is nearly start to finish how many weeks?
7 weeks.
7 weeks, 49 days, nearly 50 days, correct? Right. What is the next statement telling you? Read that statement.
The formation and development of sperms is continuous and inexhaustible in adult human males.
Continuous and inexhaustible. By the word inexhaustible, you know what they are trying to convey you? It will never stop. Understood? Understood?
Yes, sir.
What is meant by inexhaustible?
It will never stop.
It will never stop. It will never end. That is inexhaustible. Right? That means a man, a male, even he reaches 100 years old, he will produce sperms. Understood? And remember that ability is not there for females. In females, overproduction is within a limited age. Generally, if you meet a woman who is in her 50 years, most of the time they would have reached their menopause. Do you know the word menopause? Do you know the word menopause?
Yes.
Yes. After menopause, the woman will not get menstrual cycle. Her menstrual cycle is stopped. Her overproduction is stopped. No more overproduction. So, referring to a female, can we say overproduction is inexhaustible?
No.
No. In females, overproduction is in a limited age duration only. You can notice overproduction. Okay. What is the next statement?
Cell division and maturation during gametogenesis occur throughout the seminiferous tubules.
That is within the seminiferous tubule. Okay. Throughout cell division. Right. Next statement.
Hundreds of millions of sperms are produced each day by spermatogenesis.
Understandable, correct?
Yes, sir.
Next statement.
In contrast to oogenesis, in spermatogenesis, all 4 cells produced during meiosis of each spermatocyte develop into mature gametes.
Right. So the point here is one spermatocyte will produce how many spermatids?
4 spermatids.
4 spermatids. Every spermatid will turn to a sperm. Remember, one ova mother cell, oogonium. One ova mother cell, otherwise called, what is it?
Oogonium.
Oogonium. From it, though 4 cells develop, only one will turn to ovum. Understood?
Yes, sir.
That is why they say it is contrast to oogenesis. Oogenesis means whose production? Ova production. Spermatogenesis means whose production?
Sperm production.
Sperm production. Oogenesis means ova production. Oogenesis mean, what is it?
Ova production.
Ova production. In oogenesis, will all the ootids turn to ova?
No.
No. But here, all the spermatids will turn to sperms. That means from one oogonium, how many ovum you will get? From one oogonium, how many ovum you will get?
1.
One. From one spermatogonium, how many sperms you will get?
4 sperms.
4.
4 sperms. Okay. Oogenesis, will it be continuous throughout the life?
No.
But spermatogenesis?
Continuous throughout the life.
Okay, later we can write the differences between oogenesis and what is it?
Spermatogenesis.
Spermatogenesis. Okay. So spermatogenesis starts in which stage in a person?
Puberty.
Puberty. Like females, in males also there is, what is it? Puberty. Okay. Only one thing, male puberty cannot be easily recognized from outside. Due to that reason, we don't celebrate male puberty. Understood? If a girl reaches puberty, we wear her new dresses, wear jewels to her. And there's a system, right? That system is not there for males. So we don't recognize them. But this is another story I want to tell you. In which stage spermatogenesis starts in a male? At puberty. But in a female, it starts before her birth. Understood? Oogenesis in the female starts in which stage?
Before birth.
Before birth. Okay, if a female child, when she is developing inside her mother, when she is a fetus, in her ovary, it would be starting. Clear?
Clear, sir.
Right. So spermatogenesis produces mature sperms from precursor cells. Those precursor cells are at the moment to you spermatogonia. Okay, spermatogonia. They are before, if it is there, if it is called, they will be called stem cells. We'll see. Can you see spermatogonial stem cells here? Spermatogonial stem cells.
Yes, sir.
Right. Spermatogonial stem cells will undergo repeated mitosis and will produce home. Spermatogonial stem cells are producing home.
Spermatogonium.
Spermatogonium. They say spermatogonium, okay, is the one, okay, spermatogonial stem cells are already in the male fetus. Spermatogonial stem cells are already in the fetus. Spermatogonia are made from the spermatogonial, stem cells. What type of cell division is happening from spermatogonial stem cells to spermatogonium?
Mitosis.
Mitosis. Spermatogonium to primary spermatocyte.
Mitosis.
Mitosis. Primary spermatocyte to secondary spermatocyte.
Meiosis.
Meiosis 1. Correct.
Yes, sir.
Primary, secondary spermatocytes to spermatids.
Meiosis 2.
Meiosis 2. Final outcome is spermatid. Will there be cell division in the spermatid?
No.
No, but there will be differentiation turning to sperms. Correct? Right. Can you read these two lines?
Sir,
Hmm.
the stem cells that gives rise to sperms arise from mitotic division and differentiation of primordial germ cells in the embryonic testes.
Hmm.
These stem cells are situated near the edge of the seminiferous tubules.
Hmm. That is happening in which stage? That is happening in which stage? The first two line is regarding which stage? When the male is an embryo. Correct?
Yes, sir.
Before he is born. Am I right?
Yes, sir.
Rest of the story is telling after his puberty. Mature testes. Mature testes, what are the changes going on? You can read and see what I have told will be there. Read and see this.
Done, sir.
Are you clear?
Yes, sir.
Do you agree with the statement spermatid differentiate into sperms?
Yes, sir.
Yes. In a sperm, you will notice three pieces. What are the three pieces?
Head, midpiece, and tail.
Midpiece and tail. Okay. We will later discuss about it. Right. Sperms are released into the fluid-filled lumen of the tubule. They travel along the tubule into the epididymis. In the epididymis, they will become mature. They will gain the mobility. Agree?
Yes, sir.
Right. After puberty, these cells, what are these cells?
Leydig cells.
They are secreting a hormone. What is the hormone?
Testosterone.
And that hormone has a contribution on the, what is it?
Spermatogenesis.
Okay, we will later, they just say it has a promotional work on the, what is it? Spermatogenesis, correct? Right. Do you know the statement Sertoli cells are attached to various stages of the spermatogenesis?
Yes, sir.
Yes. What does the Sertoli cells do? They are giving
Nutrition to the
developing
sperm.
stages of spermatogenesis, correct?
Yes, sir.
Do you agree seminiferous tubules, from the wall of the seminiferous tubule, Sertoli cells are extended to the lumen of the tubule, towards the center of the tubule? Do you
Yes,
agree with
sir.
it? Yes. I told you already, they are secreting a hormone. What is the hormone?
Inhibin.
Inhibin. It is going to reduce the spermatogenesis. Who is the hormone promoting the spermatogenesis?
Testosterone.
Who is the hormone inhibiting the spermatogenesis?
Inhibin.
There you can see the connection, correct? Here I have selected this picture to describe the human sperm. Human sperm is a single cell. What are the three main parts of the human sperm?
Head, midpiece, and tail.
Right. What is the component located between the head and the midpiece?
Neck.
In the neck, you will see, okay, picture la mark pannunga. In the neck, you will see a pair of centrioles. What will be there?
Paracentry.
Say a pair of centrioles will be there. WW. இப்படியெல்லாம் சொல்லாம சொல்றதுகள்.
Done, sir.
Right. In the head region, Okay, surrounded by the plasma membrane, there are two components. What are the components in the head region? Tell me.
Nucleus and acrosome.
Acrosome. Acrosome, in those days we were teaching the children, is a modified lysosome. Who it is?
Modified lysosome.
Lysosome. Thing it is, in the acrosome also there are hydrolytic enzymes. In the lysosome also there are hydrolytic enzymes. The difference is the hydrolytic enzymes in the acrosome is very, very, very special. Understood?
Yes, sir.
Right. Nucleus, if you consider, in this nucleus, how many chromosomes you are expecting? A male sperm nucleus.
One.
How many? How many? It's a male sperm nucleus. In that nucleus, how many chromosomes you are expecting? How many chromosomes our father gave us?
23.
23 chromosomes. One side, those chromosomes are donated to the new life made. Other side, those chromosomes are controlling the activities of the sperm. Clear? Are you
Yes,
clear?
sir.
Good. In the neck region, there is a pair of centrioles. We will discuss about it later. In the middle piece, what are the main component there? What are the main component there?
Mitochondria.
Mitochondria. Having large number of mitochondria, what does it tells us?
It is
Sperm movement is a very active process. If you take this portion, it is called axial filament. What is called? What it is called?
Axial filament.
You know, in a flagella, there is a 9+2 structure, no? Okay. That 9+2 structure will be there in this axial filament. It is traveling through the it is passing through the tail. This tail is basically, this tail is a what kind of unit? Basically, this tail is a flagellum. Flagellum means 9+2 arrangement, correct? Understood?
Yes, sir.
Okay, this axial filament will continue through the tail to the end. Understood? Are you clear what I told you? Are you clear what I told you?
Yes, sir.
Good. So, please read the content you are given regarding the content here. If we are okay, inshallah.
Clear, sir.
Clear?
Yes, sir.
Okay, okay, okay. So, after that one, we speak about which heading, semen. What is it?
Semen.
Semen. If you take semen, it's a white color liquid that is ejaculated, released through the urethra in a male. Okay, it will be ejaculated through the urethra of a male, that is semen. If you take the semen conformably, there will be ovum. Who will be there? Sperms will be there. In addition to sperms, those glands are there, no? Seminal vesicle, correct? Prostate gland, bulbourethral gland, their secretions are also in it. At a time, okay, 2 to 5 milliliters of semen will be ejaculated. How many milliliter? How many milliliter?
2 to 5 milliliters.
In one ejaculation. Okay. 2 to 5 milliliters. In the semen, how many sperms will be there? 40 to 100 million sperms will be there in 1 milliliter. 40 to 100 sperms will be there in 1 milliliter. In that range, if the sperms are there, that man is healthy. Understood? Now say some couples after getting married, when they don't get children, they go and check is there any problems in the bodies of them. Okay. Sometimes they will be thinking with all the hardship after getting the children, they will be thinking இவனை பெற்றதை விட பெறாம இருந்திருக்கணும் அப்படின்னு சொல்லி அப்படியும் சிலதுகள் நடக்கிறது. But generally, when a couple get married, the next interest is making their own children. In that case, we will be checking the semen of the man. We'll be checking the semen of the man. If the semen doesn't have sperms or have sperms less than 40 million per milliliter, what is the meaning? No sufficient sperms. Understood? No sufficient sperms. Okay, then he will be considered as impotent, unable to become a father. Are you clear?
Yes,
Right. In the total semen, now if we take 1 milliliter semen, the portion of sperm will be only 0.1 milliliter. Understood? If total semen is 1 milliliter, in that 1 milliliter semen, how many milliliter will be sperms? 0.1 only. Okay. Other things are the one rest. In the other things, what are the components present? Mean all these are present. In the other one, mucus will be there. Certain enzymes will be there. Some chemicals called prostaglandins will be there. Vitamin C, ascorbic acid will be there. Citrate will be there. Fructose will be there. Okay. What are the materials? Mucus, some enzymes, a chemical called prostaglandins, ascorbic acid mean vitamin C, citrate, citric acid, and fructose will be there. Okay, right. Semen basically, okay, it's an alkaline material. That means pH above 7. Understood? What is the pH of semen? Okay, above 7, 7.2 to 8, like 7, 8, near to 8. So what kind of material is semen? Is it a basic material or an acidic material? Tell me. What kind of material is semen? Hello, can you hear me?
Yeah, yes, sir.
So what is the pH of semen?
7.2 to 8.
8. If the pH of semen is 7.2 to 8, okay, what kind of material it is? Acidic or basic? Acidic or basic?
Basic.
Basic. Female reproductive tract, her vaginal area is acidic. What kind of region is female reproductive tract?
Acidic.
Acidic. If sperms come to an acidic place, the sperms will be killed. Understood?
Yes, sir.
But anyway, semen should be released to the female reproductive tract. Correct?
Yes, sir.
So, this alkaline component, okay, this alkaline component is neutralizing the acidity of the female reproductive tract. Then no harm to the sperms. Understood?
Yes, sir.
Right. What is the life expectancy of the sperm?
48 to 72 hours after ejaculation.
After ejaculation, okay, after ejaculation in the female reproductive system. Understood?
Yes, sir.
So within 72 hours, it has to meet the sperm. Are you clear?
Yes, sir.
Okay, inshallah. Tomorrow we will continue that. Okay, continuously do the works. There are many works in the row. எனக்கு எல்லாத்தையும் செஞ்சு கொள்ள உண்மையில டைம் இல்ல. ஆனா செஞ்சு கொண்டு இருக்கேன். கொஞ்சம் கொஞ்சமா முடிஞ்சு கொண்டு வருது. முடிச்சு கொண்டு வாரேன். ஸ்ட்ரக்சர் பேப்பர்ஸ் ரெடி பண்ணிக்கிட்டு இருக்கேன். எம்சிக்யூ பேப்பர்ஸ் ரெடி பண்ணிக்கிட்டு இருக்கேன். ஒரு ஆள் செய்யறதால தனியா சிங்கிள் மேன் வேலையே எல்லாம் நான் தான் செய்யணும். Okay.
Okay, sir.
Right. So, inshallah, we will meet tomorrow. Bye.